So we think it opens up a broader possibility range of opportunities for our payload, whether it's on a TROP2 ADC or other antigen targets. We again think that's really helps us stand out in the place of ADC because of this unique payload and its unique mechanisms of action.
Welcome back, and thank you for joining us for another Virtual Investor What This Means segment. My name is Janine Thomas. I am CEO of JTCIR, and I will be the moderator for this segment. Today, we are excited to welcome Akari Therapeutics to our Virtual Investor platform. And I am very pleased to be joined by Abizer Gaslightwala. He is Director, President, and CEO of Akari Therapeutics. Welcome back, Abizer.
Great. Thanks, Janine. Thanks for having me here again.
Always happy to have you. And before we get started, I just want to inform our audience that Akari Therapeutics is listed on NASDAQ and trades under the ticker AKTX. And during today's discussion, the company will be making forward-looking statements. And I encourage everyone to view the company's website at akaritx.com or the SEC's website for their latest filings and information. Okay, Abizer, we're going to dive right in. So in your recent press release, you emphasize your ongoing research into how PH1 may inhibit key oncogenic drivers. So can you walk us through what prompted this deeper exploration and what questions you're hoping to answer?
Sure. Yeah. Thanks, Janine. It's a really exciting topic for us as we think about our novel payload PH1. Let me give background and reference. We have two modes of action. Again, it's a traditional cytotoxic therapy that kills the cancer cells, but we also think it has this really unique mechanism that's immuno-oncology like in terms of it activates the immune system to create broader killing of the cancer cell. So in addition to that, we're finding in certain tumor cell biologies that the PH1 payload, because it modulates a spliceosome, has unique impact and effect on the cancer cells because of that. Because some of these cancers are actually dependent on the spliceosome for some of their growth and some of their survival. So we're finding in certain tumors we actually have a different additional effect on top of the first two I mentioned that leads us to explore, well, which specific tumors, what type of histologies, what type of tumor types. So that's really exciting because that's a third avenue of biology we're uncovering around this PH1 payload.
So given PH1's unique mechanism as a spliceosome modulator, how could this research expand the therapeutic reach of your ADC platform beyond current targets like TROP2?
Sure. So I think what we're finding is because the PH1 payload has this potentially unique other ways of acting on these particular oncogenic drivers, it's interesting because then we can find tumors that are dependent on this biology and go after them, whether it's TROP2 or whether it's other antigens as well. So in particular, we found some of this, which we've already patented, we found an effect on PH1 on very well-known oncogenic drivers such as KRAS and BRAF. Those are two well-known oncogenic drivers in the space. There are many therapeutics that are approved to treat those mutations in cancer cells. We're finding not only did our PH1 payload have an effect in some preclinical tests on those types of cancer cells, but like I said, we're doing some current experiments that are going even broader than that and looking at some other interesting tumor types that are driven by certain oncogenic drivers. And if we find that's the case, we can take whether it's a TROP2 ADC or a novel other ADC with an antigen target and build an ADC that can target some of these tumor types. So it really gives us a lot more applicability and generality for taking our PH1 platform and expanding the possibilities.
Vision the potential of PH1 in tumors driven by mutations like KRAS or FGFR3, and what would you need to see in preclinical data to prioritize those opportunities?
Sure, I think again that's the beginning of the tip. We are looking at some other interesting areas that hopefully we can disclose some of that data in the near future. But if we were to take this forward, this is done at what we call an in vitro level. So I think the next step would be going to an in vivo level, looking at animal models. And if we see continued good activity either with our current ADC or we think about other targets that we want to think about, we would then progress that forward to animal testing and then even further beyond that. So yes, we're still at the early stage, but what looks really promising, in particular, how many different types of these oncogenic drivers our payload works in, which again opens up a whole new avenue of biology for us in this payload.
So, Beezer, most ADCs rely on topoisomerase or microtubule inhibitors, which have well known limitations. How do PH1's mechanisms as a spliceosome inhibitor offer a fundamentally different approach to killing cancer cells?
Yeah, again, as I think we expanded now, not only in addition to the cytotoxic and the immune activating function, immuno-oncology properties of the PH1 payload, this additional research is actually opening up new possibilities of directly affecting these oncogenic drivers. So think about another third way of affecting cancer cells, and we would say that's very broad, broader than other payloads, like you mentioned, those other two classes. We think our modes of action are multiple, and so that's what's needed to attack cancer, different ways to get at those cancer cells. So we think it opens up a broader possibility and range of opportunities for our payload, whether it's on a TROP2 ADC or other antigen targets. We again think that's really helps it stand out in the place of ADC because of this unique payload and its unique mechanisms of action.
Well, certainly exciting times ahead for Akari Therapeutics. Can't wait to have you back on. And with that, this concludes the Virtual Investor What This Means segment featuring Akari Therapeutics. I'd like to thank Abizer for joining us today. And I'd also like to thank our audience for tuning in. As a reminder, Akari Therapeutics trades on NASDAQ under the ticker AKTX. And if you like what you saw today, I encourage you to visit akaritx.com for more information on the company and to sign up to follow the company to receive their alerts as well as follow their social channels to stay current on the latest information. And you can also visit virtualinvestorco.com for our latest segments and events calendar. Abizer, thanks again. Thanks again to our audience, and I wish everyone a great rest of your day.